Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 5 for June 2023. Let's begin with the first question. Question 1. The human eye responds to different light intensities. The table shows the mean pupil diameter in different light intensities. So we see in bright light, in normal room light, and in darkness. We can see in bright light it's smaller, then it a little bit increases in normal room light, and then in darkness it becomes the biggest. They say calculate the percentage increase in the mean diameter of the pupil when a person moves from bright light into darkness. So the increase is going to be 6.41 minus 3.35, which gave us 3.06. So percentage increase will be this, divide by that, times 100, giving us 91.34%. In part B, during the fight of light response, the pupils of a person increase in diameter, which is the name of the hormone involved in the fight of light response. That hormone is adrenaline. In the next part, they say describe how this change in diameter is brought about by the muscle of the iris. Adrenaline will stimulate the sympathetic neurons, and this is going to lead to nerve impulse transmission through the motor neuron to the muscles of the pupil. So the radial muscles will contract and the circular muscles are going to relax. So this brings us to the end of question 1. Let's continue to question 2. In question 2, many of the metabolic processes taking place in cells require ATP. The electron transport chain is involved in the production of ATP. Where is the site of the electron transport chain? This is found in the inner mitochondrial membrane. The next part says enzymes are involved in the production of ATP by the electron transport chain. Which type of molecule is an enzyme? An enzyme is a protein. Next, they say describe the role of reduced NAD in aerobic respiration. And again, remember, reduced NAD carries protons as well as electrons towards the electron transport chain. This should be D. So I said reduced NAD transports hydrogen ions and electrons to the electron transport chain so that energy released as the electron transition can be used to produce ATP in the electron transport chain. In part B, carbon monoxide and cyanide affect the production of ATP. Carbon monoxide binds to the heme group of hemoglobin. Explain how carbon monoxide will affect the production of ATP. We know that hemoglobin transports oxygen. However, if carbon monoxide binds onto the heme group, then that hemoglobin cannot transport enough oxygen for aerobic respiration to occur. So I said oxygen is the final electron acceptor in the ETC, meaning electron transport chain. If carbon monoxide binds to the heme group, it will prevent oxygen from binding to that group, so hemoglobin will carry less oxygen, and there will be less final electron acceptor, so less ATP will be produced. Here they say cyanide stops the transfer of electrons along the electron transport chain. Explain why cyanide does not affect the production of ATP from anaerobic respiration. And again, this is anaerobic respiration where oxygen is not needed as the final electron acceptor, so I said anaerobic respiration is only through glycolysis in the cytoplasm of the cell. That does not involve the electron transport chain in the mitochondria. So this brings us to the end of question 2. Let's continue to question 3. Question 3, the ventilation rate of animals will depend on many factors. State 2 lung conditions that affect ventilation rate of a human. There are so many lung conditions: there is cystic fibrosis, bronchitis, emphysema, lung cancer, tuberculosis, asthma, and so many more. Just choose 2 of those and then you'll be fine. In part B, they say the table shows the mean resting ventilation rate of 5 species of animals. From the table, we see the 5 different species, we see their corresponding body masses, and we see their resting ventilation rate in beats per minute. There is standard deviation included, so the experiments were repeated, a mean was calculated, and so on. For example, using this data here, 0.54 plus 0.09 gave me 0.63, and 0.54 minus 0.09 gave me 0.45. So I showed you if there is overlap or not, and we saw for most cases there was overlap within some data sets, and that shows there is no significant difference in the data of the different animals, even with different masses. So the first question says comment on the relationship between the mean resting ventilation rate and the body mass of animals. Actually, here we see as the mean body mass increases, the resting ventilation rate decreases. So I said from the table, as body mass increases, the resting ventilation rate decreases, and this shows a negative correlation. This is a correlation and not necessarily a causation. Also, the experiment used a smaller sample size, meaning only 5 species, so that is not representative enough. There is no significant difference between some data. For example, for taller point monkey, the lowest data is 0.45, the highest is 0.63, and the ring-tailed lemur 0.49 and 0.57. We can see there is overlap within these data sets, and therefore for the different species, there is no significant difference in that data. So I say there is significant difference between the data of taller point monkeys, whose range is that, and the hippopotamus lemur, whose range is that. Now, because there is no overlap among these data sets, we can say there is significant difference. And again, for some data sets, no significant difference, while for others, there is significant difference. Next, they say the tidal volume for a resting taller point monkey was found to be 40 centimeters cubed. The minute ventilation is the volume of gas inhaled or exhaled from the lungs in 1 minute. Calculate the respiratory minute ventilation for a resting taller point monkey and give your answer in decimeters cubed to 2 significant figures. From the information, the tidal volume in decimeters cubed is going to be 40 centimeters cubed divided by 1,000, giving us 0.04 decimeters cubed. And the breaths per minute is going to be 0.5 times 60, giving us 32.4. And again, here it's per minute. So minute ventilation is going to be that times that, giving us 1.3 decimeters cubed to 2 significant figures, and that was my answer. Next, they say an increase in activity level results in a change in the ventilation rate. Describe why the change occurs and how it is controlled. We know when activity increases, there is going to be more aerobic respiration, carbon dioxide concentration is going to increase, there is going to be pH decrease, and then the chemoreceptors can detect the change, and then the nervous system can send impulses to cause increase in the ventilation rate. So I said when activity increases, the rate of aerobic respiration also increases and more carbon dioxide is produced. Increase in CO2 concentration is detected by chemoreceptors in the arteries, and impulses are sent through the sensory neurons to the inspiratory center in the medulla. From there, impulses are sent through the sympathetic nerve to increase the contractions of the diaphragm muscle as well as the intercostal muscles, and this increases the ventilation rate. So this brings us to the end of question 3. Let's continue to question 4. In question 4, movement in mammals results from interactions between muscles, tendons, ligaments, and bones. How many of the following statements about tendons are correct? Tendons act as extensors, that is wrong. Tendons are myogenic, that is wrong. Tendons connect muscles to bones, that is correct, so it's going to be one statement. In part B, the photograph shows part of a skeletal muscle fiber as seen using an electron microscope. So this is part of the muscle fiber, and they've given us a scale corresponding to 1 micrometer. They say which pair of letters represents the length of 1 sarcomere. 1 sarcomere is going to be from here to here, which is about P to U. So I chose P to U, and B is my answer. Next, they say calculate the magnification of this image and give your answer in standard form. So we are going to use this scale here. When I measured, I got 1.54. And again, you have to use your ruler. I was using my iPad, so you could use the actual paper. Some get 1.55 and so on, but I got 1.54. That is in centimeters. And we know this is corresponding to 1.54 times 10 power 4 micrometers. So my magnification is going to be the image size, which is that, divided by the object, giving me 1.54 times 10 power 4 as my answer. Next, they say describe how the structure of fast-twitch muscle fibers differs from slow-twitch muscle fibers. Fast-twitch muscle fibers are involved in intense activity, and slow-twitch muscle fibers are involved in enduring exercises. So due to that, fast-twitch muscle fibers have more glycogen to provide the glucose for anaerobic respiration. The fast-twitch muscle fibers are larger in size, of course because of the packing of the many. They are larger in size. Fast-twitch muscle fibers have fewer mitochondria because there is less need for aerobic respiration, and fast-twitch muscle fibers have less myoglobin, and again there is less need for aerobic respiration. Fast-twitch muscle fibers have fewer blood capillaries, and again they do not need oxygen to be delivered that much for respiration to be carried out, so the nutrients are going to be got from glycogen, which is already stored there, so there is no need for much capillary networks within the fast-twitch muscle fibers. In part C, the graph shows the relationship between the force of muscle contraction and the velocity of muscle contraction. So we see as the velocity of muscle contraction increases, the force of contraction decreases. They say the greater the number of cross bridges formed between actin and myosin, the greater the force of contraction of a muscle. Explain the relationship between the force and velocity of contraction of the muscle. If they ask questions to explain, you begin by stating the relationship, because here we're explaining the relationship between, meaning talk about the relationship and give a reason as to why the relationship is the way it is. So always the first mark is given for you describing what they want you to explain. So I said, as velocity of contraction increases, the force of contraction decreases. That is 1 mark. And then I can begin my explanation. Remember, with every contraction, there is going to be an actin-myosin bridge that is going to be formed. So if the velocity increases, there is going to be more actin-myosin bridges formed per second. That means the duration of the actin-myosin bridges is going to decrease. So I said when the myosin head binds onto actin, an actin-myosin bridge is formed. And when ATP binds onto the myosin head, the bridge is broken. Increasing the velocity of contraction leads to faster binding of ATP onto the myosin head, and this decreases the number of cross bridges formed per time, leading to decreased force of contraction. This is going to decrease the number of cross bridges available per time. I'm not saying the speed of their formation, but available per time. I hope this part, when I put per time, is not confusing. I mean, at one given time, observing the whole muscle, there will be fewer actin-myosin bridges, but the rate at which they're coming and going is still going to be fast because you're increasing the velocity of the contraction. So this brings us to the end of question 4. Let's continue to question 5. Question 5, the kidney is an organ that is involved in osmoregulation and the production of urine. Which substances are filtered from the blood in the Bowman's capsule of a healthy individual? So, of course, there is no filtration of prothrombin, there is no glycogen, there is no prothrombin. It should be glucose and urea, and my answer is B. Next, they say which part of the Bowman's capsule prevents plasma proteins from being filtered out of the bloodstream? That should be the basement membrane. And the next part says which transport mechanism is responsible for the uptake of glucose into the cells of the proximal tubule. This is not endocytosis, not exocytosis, not osmosis, but sodium co-transport. Part B, describe how the loop of Henle acts as a countercurrent multiplier. The descending loop is permeable to water, the ascending loop is not permeable to water, and these two loops are moving in opposite direction. So the ions or some ions can enter the descending loop, water leaves the descending loop, and then as it reaches the lower part of the loop of Henle, it's going to be hyper-concentrated, or we can say it's going to be hypertonic in relationship to the surrounding fluids. And then as the filtrate goes through the ascending loop, there is going to be more ions lost. And then at the end of the ascending loop, it's going to be hypotonic to the surrounding fluid. So I say that descending and ascending loops transport the fluids in opposite direction. Chloride ions pumped out of the ascending limb by active transport, and sodium ions follow. This is causing the fluid within the ascending loop to become hypotonic at the end of that ascending loop. This increases the ion concentration in the fluid surrounding the loop. Since the descending loop is permeable to water, water leaves it by osmosis, so the filtrate is hypertonic as it reaches the bottom of the loop. And the ascending loop is impermeable to water, so water does not leave it, yet it transports ions out by active transport. I'm talking about the loop. This creates a hypotonic filtrate as it enters the distal tubule. In part C, in an investigation, one group of people was given nothing to drink, a second group was given water, and a third group was given 1% salt solution to drink. The mean rate of urine production was recorded for the males and females in each group. The graph shows the results of this investigation. So on the horizontal axis, we have the types of drinks that were given. Under the vertical axis, we have the mean rate of urine production. Those given nothing to drink produce less urine. Those given water to drink produce more urine because, of course, water diluted the water potential of the blood. And then those given 1% salt solution produce urine more than those who had nothing to drink. So, and again, the salt solution contains some water that caused a higher water potential within the blood, and some water had to be removed. So in the first part, they say calculate the ratio of the mean rate of urine production for the females in the three groups. They want you to express your answer as nothing to drink, to water to drink, to salt solution to drink. For the females, 100. This corresponds to 840, and that is to 440. So the ratio is 100 to 840 to 440, which is approximately 1 to 84, and that was my final answer because they wanted to one significant figure. Next, they say describe two conclusions that can be drawn from the results of this investigation. From the investigation, we can see that drinking leads to production of more urine. No matter what they drank, there was more urine produced. And from the results, the females produce higher volume of urine in comparison to men after a drink. Next, they say explain the effect on the mean urine output of the group drinking water. The effect is drinking water increases the water potential of the blood, so ADH hormone is not released from the pituitary gland, and then the collecting ducts and the distal convoluted tubule will be less permeable to water, less water reabsorption occurs, and the volume of urine produced is going to increase. This is the explanation for the effect that we saw. Drinking water causes production of more urine, and this is the explanation for that. So this brings us to the end of question 5. Let's continue to question 6. Question 6, the concentration of ions in the blood is controlled by homeostasis. Explain how ions can cross the membrane. Ions cannot cross the membrane directly through the phospholipids, so they can go through channel proteins as well as carrier proteins. Carrier proteins transport both by active and passive transport, while channel proteins transport only by passive transport. So here I said ions are charged, so they can't cross the membrane directly through the phospholipids. The cell membrane contains channel proteins and carrier proteins. Then ions are transported across the cell through channel proteins or carrier proteins using facilitated diffusion from high concentration to low concentration, or they are transported via carrier proteins by active transport against concentration gradient using ATP. In part B, ions are found within fluids in the human body in different concentrations. The graph shows the concentration of ions in intracellular fluid, which is the cytoplasm, extracellular or tissue fluid, as well as the blood. So here we have the different ions. There is sodium, there is potassium, chloride, and hydrogen carbonate, and their corresponding concentrations in the cell, outside the cell, and then in plasma. Potassium is the highest in the intracellular fluid, and of course all these ions are high or lower in different fluids for certain purposes that they carry out. The ions are used to perform vital functions within the metabolism of the cells. Discuss how the distribution and concentration of ions shown in the graph contribute to their biological roles. Use the information given in the graph and your own knowledge to support your answer. So I began with potassium. Potassium is the highest in the intracellular fluid, but have almost the same concentration in the extracellular fluid and blood plasma. This is because potassium ions activate some enzymes. They are also useful in nerve impulse transmission. We can see this is almost the same, but here it's going to be the highest. And for sodium, the ions are high in both blood plasma and extracellular fluid. Sodium is useful in water balance in the body and nerve impulse transmission. When we go to chloride, chloride ions are high in the extracellular fluid and in blood plasma compared to the intracellular fluid. Chloride ions are involved in water balance. They're involved in production of HCl. There is hyperpolarization of neurons and so on. Then the hydrogen carbonate ions are low in intracellular fluid and a little high in extracellular fluid and blood plasma. This is because the hydrogen carbonates are useful in the buffering of pH of the blood during transport of carbon dioxide from the tissues towards the lungs. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Serotonin is produced by neurons in the brain. How many of the following statements about serotonin are correct? The first one, L-DOPA can be converted into serotonin in the brain. That is wrong. Serotonin is a neurotransmitter. That's correct. Serotonin is produced by postsynaptic neurons. That is also wrong. So only one statement is correct. In part B, they say there are 14 different serotonin receptors found in the human nervous system. 3 different genes are used to produce these receptors. Explain how 3 genes can produce 14 different receptors for serotonin. And again, remember these receptors are proteins, so if 3 genes are used to produce 14 different proteins, it means there is some post-transcription modification that occurs. Introns are cut out, the exons are rearranged in different orders in order to create the multiple messenger RNAs that are mature for translation into different proteins. There is also a possibility of post-translation modification. So I say these regions are activated, and this leads to transcription to produce pre-messenger RNA. The pre-messenger RNA goes through post-transcription modification where introns are removed, and the exons can be rearranged to produce 14 different mature messenger RNAs. The mature messenger RNAs leave the nucleus and are translated in the cytoplasm. Many different amino acid sequences are made. Also, after translation, modification could occur where proteins are shortened or lengthened, and this leads to production of more proteins, or these proteins act as receptors for serotonin. In part C, they say the effect of MDMA, which is ecstasy, on serotonin concentration in rats was investigated. The rats were given different concentrations of MDMA by mouth twice a day for a week. The concentration of serotonin in the cerebrospinal fluid of the rats was analyzed 24 hours after the last dose of MDMA. Cerebrospinal fluid is fluid that surrounds the brain and the spinal cord. The graph shows the effect of MDMA on the concentration of serotonin, and the line of best fit is shown on the graph. So we see the concentration of MDMA and then the concentration of serotonin. Overall, meaning following the line of best fit, the higher the concentration of MDMA, the higher the concentration of serotonin produced. So here they said determine the effect of MDMA on the serotonin levels in the cerebrospinal fluid. It said overall, as the dose of MDMA increases, the concentration of serotonin increases. From 0 to 5 milligrams per kilogram MDMA, the serotonin levels increased by 70 milligram per centimeters cubed. You can refer this from the graph. Predict the concentration of serotonin for a dose of 17.5 milligram per kilogram of MDMA. They say use the line of best fit and give your answer in gram per centimeter cubed. I extended my line of best fit to about 17.5 and get about 400 milligrams per centimeter cubed. So this is going to be 0.4 as my answer. Here they explain how the MDMA taken by mouth could affect the levels of serotonin in the brain. Use the information in the question to support your answer. If it's taken by mouth, digestion is going to occur into smaller particles that are absorbed into the blood. Then they cross the blood-brain barrier in order to cause their effect. So I said MDMA is digested and absorbed into the bloodstream. It will then cross the blood-brain barrier. It will bind to the receptors in the brain, and this will stimulate the release of serotonin in the brain. So this brings us to the end of question 7. Let's continue to question 8. Now, before we go to question 8, I'm going to begin by showing you the scientific article. I'm going to be silent, and then when we begin the questions, I'll be able to walk you through them. So this is a scientific article for June 2023. So let's look at question 8. They say the scientific document you have studied is adapted from the article Microbiota, Gut-Brain Axis, and the Central Nervous System. They say use the following information from the article and your own knowledge to answer the following questions. Part A says gut microorganisms play an important role affecting human metabolic function by decomposing the complex polysaccharides in food. Explain the role of gut microorganisms in decomposing complex polysaccharides. Now, these are complex polysaccharides that our enzymes cannot break down. It means these microorganisms can produce or release enzymes that will digest these complex polysaccharides. So I said humans can produce enzymes that can digest complex polysaccharides. However, the microorganisms release these enzymes to carry out this digestion, and the products of digestion can be absorbed into the human body through the villi. In part B, describe how the vagus nerve is involved in the control of heart rate. The vagus nerve includes nerves of the parasympathetic nervous system. So, of course, controlling heart rate begins with chemo and baroreceptors. I said chemoreceptors and baroreceptors in the aorta and carotid bodies detect change in pH and pressure changes in blood. They send impulses through the sensory neurons to the cardiovascular control center in the medulla, and then impulses are sent through the parasympathetic nervous system, part of the vagus nerve to the sinoatrial node. This slows down the rate of depolarization of the sinoatrial node, so the heart rate is going to slow down. In part C, microorganisms can also enhance the anti-tumor effect of drugs by promoting T-cell accumulation and transformation. Explain how microorganisms promote accumulation of mature T killer cells that enhance the anti-tumor effect of drugs. These microorganisms produce chemicals, and these chemicals are going to be absorbed into the blood, and then the immune system is going to detect them. T killer cells are going to be produced or activated, and then they will increase in number, release bufferins that will cause the destruction of those cells. So I said microorganisms release chemicals that are detected by the immune system, and they activate the T killer cells. Clonal expansion, meaning increase in the number of the T killer cells occurs, and then the T killer cells release bufferins. These destroy the tumor cells. In part D, describe how fMRI scans could be used to show the activity of the regions of the brain controlling memory and sensation when probiotics are consumed. To do this, the patients or subjects have to be given the probiotics before the scan, and then a scan can be carried out to see the regions that are activated. So I say that test subjects are given probiotics, and then fMRI scans are used. Activity of regions of the brain that control memory and sensation can be compared. In part E, suggest how changes in diet could affect the composition of the gut flora, causing imbalances in the naturally occurring chemicals in the brain. Of course, when you eat different foods, these are going to be taken up by microorganisms. Metabolites are going to be produced, and these metabolites could be different if different foods were consumed. So I said if diet changes, the gut flora will release different metabolites, and these are absorbed into the bloodstream where they cross the blood-brain barrier. Neurons will be stimulated to release different chemicals or different neurotransmitters due to the different metabolites that have entered the blood. Then they say multiple sclerosis, MS, is a demyelinating disease of the nervous system. Here, paragraph 14. Symptoms of MS include poor coordination and loss of vision. Explain how demyelination could result in these symptoms. Demyelination is removal or loss of myelin from neurons. This is going to slow down the speed of nerve impulse transmission. So I said as the neurons are demyelinated, nodes of Ranvier are decreased, meaning saltatory conduction is also going to decrease. This leads to slower speed in nerve impulse transmission. Gut microorganisms produce LPS, paragraph 15 to 17. White blood cells have receptors for LPS on their cell membranes. Describe the technique that could be used to identify the LPS receptor gene found in white blood cells. The technique is using microarray accompanied by bioinformatics, so we'll extract messenger RNA from the white blood cells, and then it could be used to make cDNA. Then microarray with specific sequences is used, and the data from the microarray can be analyzed using bioinformatics in order to identify the LPS receptor gene found in the white blood cells. So this brings us to the end of question 8 as well as to the end of this whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.